All right, have you ever stopped to think what you have in common with a huge, bustling city? Probably more than you think. Today, we're diving into a quick 10-minute tour of the most fundamental city there is, the cell. Let's jump right in.
Okay, to really wrap your head around this, I want you to picture it. You're not just you. You are a massive community of something like 37 trillion tiny cities all working together in perfect harmony. So for the next 10 minutes or so, we're going to go on a tour of one of these amazing little cities.
So, what is a cell really? Well, at its most basic, it's the fundamental unit of all life. You know how a house is built from individual bricks? It's exactly like that. Every single living thing, from a tiny little amoeba all the way up to us, is made of cells. They are the absolute basic building blocks of life. Okay, so every city has to have borders, right? And a government, or a city hall. In our little cell city, those jobs belong to the plasma membrane and the nucleus. Let's start right at the city's edge. The plasma membrane, think of it like the cell's outer skin. But it's not some dead wall, not at all. It's a living, flexible border patrol. The scientific term is selectively permeable, but honestly, that's just a fancy way of saying it acts like a club bouncer. It's super picky about who and what it lets in and out. Now, this is something that trips a lot of people up. How do things actually get across that border? Well, there are two main ways. First, you've got passive transport. This is the easy one. It's like rolling a ball downhill. It just happens naturally, no energy needed. Things simply move from where there's a lot of them to where there's less. But then you have active transport. This is the complete opposite. It's like pushing that ball uphill. You're going against the grain, from low concentration to high. And that takes a lot of energy, which the cell pays for with a molecule called ATP. Okay, let's zoom in on a special kind of passive transport called osmosis, which is all about how water moves. This is super important. If you put a cell in a hypertonic solution, there's more stuff outside, so water rushes out and the cell shrinks. In an isotonic solution, everything's balanced, so nothing really changes. But in a hypotonic solution, water rushes in and the cell swells up. And here's a great little trick to remember it. Think hypo makes the cell swell up like a big hippo. All right, let's head to the center of town, to City Hall, or in this case, the nucleus. This is the absolute control center for the whole operation. It is so important that it's protected by a double-layered security system. And inside, it holds the master blueprints for, well, everything the cell needs to do and be. So what exactly are these blueprints? Well, it's all about DNA. Most of the time, the DNA is just hanging out inside the nucleus in a messy, thread-like form called chromatin, kind of like a ball of yarn. But when it's time for the cell to divide, that yarn gets wound up super, super tight into these neat rod-shaped packages we call chromosomes. And if you look at a tiny piece of that DNA that holds the code for one specific thing, say your eye color, that little piece is called a gene. You know, having this well-organized nucleus is actually a huge deal in biology. It's the main thing that separates the two major types of cells. On one hand, you have the simple prokaryotic cells like bacteria. Their DNA just kind of floats around in a region called the nucleoid. On the other hand, you have the complex eukaryotic cells, like the ones that make up plants, animals, and us, which have that true membrane-bound nucleus and a whole bunch of other specialized departments. Okay, let's get to the part that I know can be a real headache for a lot of students. We're going deep inside the city now to meet the workforce, the organelles. Think of these as the tiny organs of the cell, and each one has a super specific job that is absolutely critical to keeping the whole city running. First on our tour is the endoplasmic reticulum, or just the ER for short. I want you to picture this as a massive, sprawling network of highways and factories all connected together, winding throughout the entire cell. Its job? To make and ship key materials. Now, the ER isn't just one thing. It's got two parts. You have the rough ER, and it's called rough for a simple reason. It's covered in little bumps called ribosomes. And since ribosomes are basically tiny protein factories, the main job of the rough ER is, you guessed it, making proteins. Then you have the smooth ER. No bumps, no ribosomes. Its main job is making lipids, fats. And it also does other stuff, like helping your liver detoxify things. So, after the ER makes all these proteins and lipids, what happens next? Where do they go? Well, they get shipped over to the Golgi apparatus. This is basically the city's post office or shipping center. The Golgi takes those newly made products, does a little modification, sorts them, and then packages them up to be sent out to exactly where they need to go. Of course, any busy city is going to produce some trash, and our cell is no different. The cleanup crew for the cell is the lysosome. Think of it as the city's recycling and waste disposal plant. They're basically little sacs filled with powerful digestive enzymes that just gobble up waste, foreign invaders, and old worn-out cell parts. But they also have a much darker nickname, suicide bags. And here's why. If a cell gets too damaged or old, the lysosomes can actually burst on purpose, releasing those enzymes and, well, digesting the entire cell from the inside out, a form of self-destruct. Okay, but think about all this work. Making things, shipping them, cleaning up, none of it could happen without energy. So where does the power come from? That's the job of the mitochondria. You've probably heard it called the powerhouse of the cell, and that's exactly what it is. It takes the food the cell gets and turns it into ATP, which is the energy currency that fuels literally everything else.
All right, now let's talk about a few organelles you'll only find in certain cells, specifically plant cells. These are called plastids. The most famous one, of course, is the chloroplast. These are the cell's solar panels. They capture energy from sunlight and use it to make food in a process we all know as photosynthesis. But there are other plastids too, like leucoplasts, which are basically just storage tanks for things like starch and oils.
And that brings us to the city's warehouse, the vacuole. These are essentially storage sacs. Now, in animal cells, you might find a few small temporary ones, but in plant cells, it's a whole different story. They have one massive central vacuole that can take up most of the space inside the cell. When it's full of water, it presses outwards on the cell wall, which is what gives plants their rigid structure and helps them stand up straight. So we've touched on some of the key differences already. Let's put it all together now and do a direct side-by-side comparison of our two main types of complex cities, the plant cell versus the animal cell. This right here is a perfect little summary for revision. Let's run through it. The big ones? Plant cells have a tough, rigid cell wall on the outside for support. Animal cells just have that flexible membrane. Plant cells have plastids, like chloroplasts, to make their own food. Animal cells absolutely do not. And like we just said, plant cells have that one gigantic central vacuole, while animal cells just have little ones. All of this leads to their shapes. Plants are more fixed and boxy, while animal cells are more flexible and irregular. Okay, but a city can't just stay the same size forever, right? It needs to grow, make repairs, and sometimes create entirely new cities. And for a cell, all of this happens through a process called cell division. And this brings us to another classic point of confusion for students, mitosis versus meiosis. Here's the simple breakdown. Mitosis is for everyday growth and repair. One cell divides once to make two identical daughter cells. Think of it as cloning. Meiosis, on the other hand, is only for making reproductive cells, or gametes. One cell goes through 2 rounds of division to make 4 unique daughter cells, each with half the original DNA. So, to put it simply, mitosis makes clones for repair, meiosis creates genetic variety for reproduction. You know, this whole idea of cell division really gets at one of the most fundamental rules of biology. It was summed up perfectly by a guy named Rudolf Virchow way back in 1855. Who said, all cells came from pre-existing cells. It's a simple idea, but profound. Life doesn't just pop out of nowhere. It's a continuous chain, passed on through this amazing process. And with that, our whirlwind tour of the cell city comes to an end. I hope you can see now that a cell isn't just some boring blob of jelly. It's an incredibly complex and organized little metropolis, where every single part has a vital role to play. Which leaves us with one final and really important thought. What do you think happens to the whole city when just one of those tiny workers goes on strike? The answer to that question, it turns out, is the key to understanding what goes wrong in so many diseases. Something to think about.